and then we start. If you have any doubts, we start with the doubts, or then we start with the class. So there are two very simple rules in the class. One is that it has to be a discussive class, so we do discussions, we study through discussions, because biology is very tedious to remember. There are lots of things to remember, and one cannot memorize all these things, but one can understand these things to an extent where the memorizing load goes down. So you don't have to memorize everything if you understand what's happening, and you can write it in your own words. So what's left to remember and memorize is specific names of bacteria, species, enzymes, pathways, etc., etc. But other things you just know how is it happening if you understand the logic. And second thing is, do not expect a monologue in the class. I'm not going to tell you a story, and you will also have to participate at some point or the other. So today it's someone who's summarizing for you, tomorrow it can be you as well, and if no one will volunteer, then I'll pick someone randomly to summarize. Okay, sounds good to you?
Okay,
Samir?
sir.
Okay, very well. So who's going to summarize today? We also have Wafia and Nabil Saifullah joining. Good evening. Nabil, would you like to summarize today? Are you there?
Yes, sir.
Yeah, okay.
So,
Let's begin.
microbes. This is the new chapter, microbes in human welfare. In that, microorganisms are. We see the benefits of microorganisms for us in a variety of ways. Some of them are in making of biofertilizers, in the food, for example, in the curdling of milk, and also in the making of cheese and some other dishes. Then the biocontrol agents or in the agriculture, in baking, in industrial production, in baking, and so similar to baking, they are also used in fermentation and production of alcohols and other beverages. Then they're used in medicines as antibiotics, and they're used in sewage treatment. They're used as gut microbes in humans as well as other animals like the ruminants, that is cows and cattle, and the biogas production, which is used for a source of energy in the rural areas. Then next one is now uses of microbes in the household products. First one is lactobacillus. This is also called as a lactic acid bacteria. Its main function is to convert milk into curd. So when a small amount of inoculum, that is a small amount of sample bacteria, is added to the milk in a small amount of time, it will initiate the curdling process, and the entire milk will be converted into curd. Then the major plus point of this is that it increases the vitamin B12 quantity in the food. Then
Yes.
thereby increases the nutritional quality of the food. The next one is the cheese. In the production of
Okay,
cheese,
okay.
we use
Just one second, Nabil. Sorry to interrupt you, but before you go forward, did I tell you how does milk becomes curd? So the proteins, how do they start coagulating? No, right? So this lactic, as I said, that lactic acid bacteria, so they start producing lactic acid. And have you seen or have you heard, or like, if you squeeze lemon in the milk also curdles very fast. So but that's so fast that the water stays away and all the protein curdles together. And now the milk is of no use. You cannot again homogenizely mix it and drink it as milk. The only thing you can do is to make paneer out of it. Have you seen this happening at your homes? No, no one. Yeah. So like in Hindi we call it that like doodh phat gaya, and the only thing you can do now is to make paneer out of it. So that happens. So in acidic environment, proteins coagulate. Many proteins, they just coagulate, come together and coagulate. They come out of their solution form and coagulate and become insoluble in the water. So but that happens slowly over time as the bacteria, in a very controlled way, in lactobacillus case. So bacteria keeps dividing in the milk because milk has lactose, so it converts it into lactic acid, utilizes it, converts it into lactic acid, and this acid makes the milk come together as a curd, okay, with the water in it. Sometimes you will see that if a milk has water, some water comes out from the curd. If you cut the curd, some water will ooze out, right? You've seen that happening or not? So it's because of the acidic components rising in the milk that the milk starts curdling together. And of course, the number of bacteria as well. Yes, Nabil, continue. Are you there, Nabil? Am I audible, people?
Yes, sir, you are.
I think Nabil is not in the meeting or what? Nabil is there. Nabil, are you there? You are mute if you are speaking. I think Nabil can't hear me. Anyone else would like to continue from here? Mehraj?
Sir,
Yeah, would you like to continue from here? I think Nabil is
I was
not able to.
there.
Okay, you were not there in the last class. Okay, Saifullah, you were there in the last class?
Yes, sir.
Yeah. Yes, please.
Yes. So the second exam is Propionibacterium shermani. It is used to make Swiss cheese.
Yeah.
The holes in this seed is due to the release of carbon dioxide from this bacteria.
Right.
And then the third one is Saccharomyces cerevisiae. It's also called a baker yeast.
So both, so Propionibacterium shermani is a bacteria that releases CO2. It ferments, so it makes the cheese through fermentation. The same process of fermentation where CO2 is released is also done by yeast, which is Saccharomyces cerevisiae. It's called baker's yeast, baking yeast, or budding yeast, and it causes fermentation in the dough, in that starch, carbohydrate food source, and as a result, that dough you get the cakes and you get the bread made. Correct?
Yes, sir.
Yes, and in the end, we discussed about microbes in industrial products. We were taking upon Penicillium notatum first thing, which is an antibiotic. So Penicillium notatum is a fungi that secretes. So let's start with today's class from here. So it secretes this compound called penicillin that kills bacteria in its presence, and this is what we still use, and the derivatives of penicillin that we use to make antibiotics and treat bacterial disorders, right? Now, antibiotic means against life. So the word anti means against. And the word biota comes from life. Now, whose life this compound is against? Our life? No, the bacteria's life, right? Are you there, people? Am I audible?
Yes, sir.
Yeah. So the bacteria's life. That's why they are called antibiotic. So antibiotics is a broader term. The kind of antibiotic penicillin is called antibacterial. There can be antiviral, antifungal. They all come under the broader class of antibiotics. But because we mostly use antibiotic to treat bacterial diseases, we just call them antibiotic. Okay, so penicillin is the first discovered antibiotic. It was discovered by Alexander Fleming. Just remember that. Alexander Fleming discovered it, and he was working. This was discovered he was working on the bacteria Staphylococcus. So this is the bacteria against which he first discovered it. Okay, Staphylococcus he was working with. He once observed, so he was actually working on bacteria, not on fungi. But he once observed, so you know how bacteria are cultured? There are Petri plates or culture plates on which there is a medium, which is mostly your like agar based medium, on which you culture bacteria. So the bacteria on that medium will look like colonies, like these black colonies, and you know that these are the bacterial colonies. Now he was working on it and he left some of these plates untouched somewhere and left from his lab, and they were left untouched for some time, and he figured out that a mold started growing on it. So something started growing on this plate because this plate has nutritive media, and in its vicinity where there should have been bacteria, now there is nothing. All these bacteria gets killed in its vicinity. So he could see that something is growing, and in its vicinity the plate does not have any bacterial colony of the Staphylococcus on which he was working. Now this was very strange. So he thought maybe this thing which is growing is secreting something which is killing the bacteria. And that's how he figured out that indeed this mold was Penicillium notatum, the fungi, and the compound that it was secreting is penicillin. He named it penicillin. Okay. But he just figured this thing out. He did not made antibiotic. So from this penicillin, there were two scientists who used penicillin as antibiotics for the first time, or who converted this discovery into a full potential antibiotic. They were Ernst Chain and Flore, Howard Flore. Just remember this name. These two first established penicillin as antibiotic. Is it clear? Ernst Chain and Howard Flore. And this was very useful during the Second World War, because for America, and many American soldiers were wounded. So earlier, before we got to know about antibiotics, even a small infection that we cannot contain. So earlier there were, you know how infections were treated? If someone had an infection in limb or a hand or because of a wound, how will you treat that infection? Leaching was one way. Yeah, leaching was one way, but it also has many problems because leach secretes many toxins in your blood instead of just
And for alcohol.
Alcohol. You know, if you can, alcohol were tried to kill many bacteria, but many bacteria do not die with alcohol. Instead, there can be toxicity because of that as well. So there's something called you heat a metal, or a metal is called it gets sanitized properly, heat, and heat can kill tissues and cells and also bacteria. So the region which is infected, if it is small, you can burn it with heated metal rod. So of course it will leave a burnt, third-degree burn scar on your skin, but it will also be, sometimes it can treat infections. So we had no other way to treat infections, and people used to die with very small infections. For example, if someone had a bacterial infection because of which they got a small wound, and slowly, if you cannot contain it, it will spread in that whole region, organ, and the person will die because of septic shock. So you know septic shock, because of bacterial infections, you can die. High fever and people used to die. So even especially in wars, when soldiers used to get wounded, because they are fighting wars in all sorts of places where in your wound you can get bacterial infection very easily, and soldiers either used to ampute that part if it is in a finger or a hand. If you want to survive, then you get the hand amputated to not let the infection spread, or the people used to die. But after the discovery of antibiotic, the Second World War, many soldiers were saved. Okay, and that was also the trial ground for antibiotics. So normally it's unethical to just trial any drug on humans, but if it's in a war situation, that soldiers are anyway going to die, right? So if they are dying, why not try penicillin? If their infection is controlled, maybe they can survive. That's a win-win situation. Anyways, they are dying because of the wound and because of the war. So because of which all the three, Alexander Fleming, Chain, and Flore, all these three were awarded Nobel Prize in 1945 post the Second World War. So Second World War was between 1939 to 1944 or 45, and in 1945 all these three people were awarded the Nobel Prize in Medicine and Physiology, okay, for this discovery. So this is another case of microorganisms working for our benefit. Apart from that, microorganisms are also used for making useful enzymes and bioactive molecules. We call any molecule bioactive if it goes inside the biological system and helps or shows some effect as a molecule. Now, for example, how many of you know about certain organic acids that are important? For example, citric acid. Citric acid?
Yes.
Where do we use citric acid? Or acetic acid? Or lactic acid, butyric acid? So all these names. So this bacteria, Aspergillus niger, it is used to Aspergillus niger is a fungi. It is used to produce what? So this is for citric acid. Where do we find citric acid normally? What is citric acid, people?
Oranges.
All the citrus fruits, orange, lime, lemon, mosambi, all the citrus families produce citric acid. So these are some acid producing microorganisms. Okay, then so this is a fungi. Another one is a bacteria. It's called Acetobacter. Now by the term you can tell me what does it produces. Acetobacter aceti. It produces acetic acid. And where is acetic acid useful? Acetic acid, where is it useful? People, no one of you likes chow mein or Chinese food? Okay, I think no one has. So how do they make chow mein or the noodles? Yes, Madhya has written vinegar, correct. What is vinegar? Vinegar is a dilute acetic acid only. Okay, it gives you that flavor, and that when you cook, we use vinegar to cook, right? Vinegar is also used to make pickles. Yes, no? Yes. So that's where acetic acid is used in food industry as well. Then there is butyric acid, and it is by Clostridium. Clostridium butylicum. The name butylicum, it produces butyric acid. Another organic acid that is used for certain drugs and medicines. And then of course we studied lactobacillus, which produces lactic acid, okay, which helps in curdling. So these are acid producing organic acid. So organic acid is also a bioactive molecule. It shows effects in the living systems. Okay. Then so these are acid producers, organic acid producers, most common examples. After that we have alcohol is another commercially required substance. Alcohol is used to make beverages, alcoholic beverages, which is like a big industry. Alcohol is also used to make different alcohols for sanitizers. You know, during the COVID, all your hand sanitizers contains alcohol. What property of alcohol makes it a useful substance? Anyone? Anyone knows what do you have in your hand sanitizers? Your hand sanitizers are mostly 70% ethanol. You know that? Okay. So yeast, which is again Saccharomyces cerevisiae, so this is used for the production of ethanol through fermentation of sugar. Now ethanol is used for making sanitizers. Ethanol is also used for alcoholic beverages, and it's also used for medicines. For medicines, many medicines are alcohol based because what property of alcohol or ethanol helps us with sanitizers and medicines? Anyone? No one knows?
Sir, can you repeat again?
What properties of alcohol is helpful in medicine industry and for making sanitizers?
Killing bacteria, germs.
Yes, it has antibacterial property. So many of our medicines as a solvent, when we have to dissolve the drug as a solvent, we don't use water. Water can be heavily infected or contaminated by bacteria. We use ethanol, alcohol, because that cannot be contaminated by bacteria. Bacteria do not do well, do not survive in alcohol. Anyone knows why? Why do they kill bacteria? How do they kill bacteria? Anyone knows? Antibacterial property. But what? How? Okay, figure it out and tell me in the next class. Why does alcohol kill bacteria and how? Okay, nice. Also, we use, how many of you have heard of in some antibiotics are used in treatment and therapies? How many of you have heard of clot busters? So people with heart conditions and the condition where clots of cholesterol, gets deposited in the blood vessels or arteries and veins, they are given streptokinase. So there is a bacteria which is called Streptococcus. We just studied in the last class one species of Streptococcus that was not useful to us, that was harmful. Can anyone tell us which Streptococcus is harmful to us? It was Streptococcus pneumonia. In the last chapter we studied that. Are you all able to hear me? So you are not participating at will, right? I should know. If you are not interested, I can just keep going on and give a monologue, but that's not how I would like this class to be. So these are all fact-based things. It will be very difficult for you to remember it, and the effort is being put so that you discuss and figure out, make links between certain things and remember it easier, in an easier way. So Streptococcus pneumoniae was a bacteria that caused pneumonia, right? Lung infection in humans. Some
Yes, sir.
Streptococcus, others, we can genetically, this is a bacteria, you can genetically modify, genetic modifications, and to produce what? Streptokinase, coming from Streptococcus, so it's called Streptokinase. And if it is a ACE, can you tell me what it is as a molecule, as a compound?
Enzyme.
Yes, ACE means enzyme. So this is used in therapy for treatment for myocardial infarction. Now tell me, the word myo means?
Muscle.
Cardiac means?
Heart.
Heart. Myocardial means muscles of the heart. Infarction means they get killed. That's called heart attack. So myocardial infection leads to heart attack. So in this, it is used for the treatment, and streptokinase is known as clot buster. Like, it's used in clot busters. So if there's a clot somewhere leading to myocardial infection and heart attack, streptokinase can help, and that we produce from the Streptococcus bacteria by genetic modifications. Okay, it's used to remove clots in this. Used to remove clots in heart. Okay, people. So it can prevent a patient from dying in myocardial infarction and can prevent heart attack. Is it clear? So this is a very bioactive. So these kind of molecules like streptokinase are called bioactive molecules. So they have an active role to play in biological systems, which can be helpful for us. Another bioactive molecule, if you have heard of it, is called chlorosporin, cyclosporin A. Have you heard of this? Cyclosporin A. Anyone has heard of this? So it is a immunosuppressant. In the last chapter, we said about organ transplantation, remember?
Yes.
And what used to happen in organ transplantation? Can anyone take me through it quickly? Whenever we get an organ transplanted from a donor, what happens to that organ? And how our immune systems, just simple in 1 or 2 lines, basic.
So the immune system does not accept the organs, so it tries to kill it.
Yes, right? So it tries to cause an infection with that organ. The organ gets rejected. So what do? We can't afford the organ to be rejected. We will die. So what do?
We use my immune suppressor.
Yes, we tell the immune system to be silent. Don't worry. We know what we have done. It is a foreign particle. It is a foreign tissue. We know, but we have deliberately transplanted it to survive. So to keep the immune system in a suppressed form, we need to give these drugs called immunosuppressants. So cyclosporin A is one type of immunosuppressant which is used in patients with organ transplantations. Okay. And this is produced by a fungus. And the name of the fungus is Trichoderma polysporum. Okay. Is it clear? So this is another bioactive molecule. Do you understand, everyone? Now, just like Streptococcus, we also have studied Trichoderma somewhere in the last class. Can anyone tell me where have you studied Trichoderma? Some Trichoderma are also not useful, they are harmful. Quickly. Which disease was caused by Trichoderma? Yes. No one is audible to me. People, which disease is caused by Trichoderma? Okay, figure out. In the last class, where have we studied this? Please don't keep forgetting. Another thing that you should know is statins. Anyone knows what is statin? Statins are another bioactive molecules which are used to lower blood cholesterol level. So someone who has very high blood cholesterol, so cholesterol is of two types, high density and low density. Low density cholesterols are mostly bad cholesterols. So an HDL, which is high density lipoproteins or high density cholesterol, is good ones. So our body needs cholesterol, not cholesterol. Sorry, cholesterol. So used to lower blood cholesterol levels. And this is produced from another fungi, which is called Purpurus. Monascus purpurus. Okay, and this is a fungi. So these are some common, and so basically what they do is there are enzymes which are responsible or which play a role in the synthesis of cholesterol in our body. So if more cholesterol is synthesized, it gets accumulated somewhere. That's where the problem of myocardial infarction can also happen. So they react with those enzymes and do not allow the formation of excess cholesterol to happen. So these statins. Okay, so statins are given, another bioactive compound. So these are the few examples that you have to study and understand. And now, where else do we use microorganisms?
So
Microbe.
bioactive molecules means what?
Sorry?
Bioactive molecules means what?
Bioactive molecules means the molecules which are synthesized by some microorganisms, or you can also synthesize it artificially in the lab, but they are active biologically. They do some biological functions. You understand?
Okay, yes.
So if I put some synthetic, there are many molecules, right? Many molecules in nature, but all those molecules inside the cell are not doing anything. Okay, for example, iron oxide or any other molecules. Iron might be useful to make blood, but if you know about any other molecule which is so you know, chemistry has physical chemistry, organic chemistry. Am I audible, everyone?
Yes.
Yeah, sorry, I lost the network. Let me share the screen. So I'm facing some issues with the network, so you all can hear me clearly, right?
Yes.
If I keep my
Yes.
camera off. Okay, good. So we were talking about this. Do you understand what do we mean by bioactive molecules? Anyone has any doubts with that? No one has any doubts?
No, sir.
Okay, so apart from making, so we are started with household, the common household. See, this is how we are, just give you a context. We started with households, you know, some common thing like curdling, lactobacillus, cheese which is used, baking yeast. Then we went to the industrial things where we studied about Penicillium notatum, which is a fungi, secretes penicillin. We use it as antibiotic. Then to make bioactive molecules like organic acids, which are used for multiple things. For alcohol production, we used again yeast, and ethanol has antibacterial property, about which you will figure out in the next class and tell me how it kills bacteria. And or I can say how it does not allow bacteria to grow in it. So it can be used for sanitizers, beverages, and medicines. Then for treatment and therapies, there is Streptococcus, through which we get Streptokinase, which is a bioactive molecule used as a clot buster to treat myocardial infarction. Or cyclosporin A, which is an immunosuppressant agent and used in given to patients with organs transplanted, and it's produced by a fungus called Trichoderma polysporum. Then statins, again by a fungus Monascus purpurus, used to lower blood cholesterol levels. Is it clear till here, everyone?
Yes, sir.
Okay. Now, something that has high chances of coming in your exams from this chapter, which is microbes in sewage treatment. This is another. Now, what is sewage basically? It is waste, human waste and all sorts of waste produced from the cities, which gets mixed with water and is thrown out from the cities through sewers, right? We clean human waste, another kind of waste, through water, right? We use water to clean it. So that contaminated water is called sewage water or sewer. So the major component of this wastewater is human waste, human excreta. Okay. So this word sewage is waste water through sewers. Okay, and the major component is human excreta, which can cause a lot of diseases. Okay, and it contains large amount of microbes. So sewage or sewer water contains huge amount of microbes, and the problem is majority of these microbes are pathogenic. Is it clear? Contains large amount of microbes and mostly they are pathogenic. Now we cannot keep throwing water from the cities, because where are we going to throw them? Where are we going to throw these thousands and thousands of tons of liters of water? Where do the sewers finally go? Where does cities sewers go? You all live in cities, right? Do you know where do your sewers go? They go into water bodies, in your rivers, right?
Yes, sir.
So if we just, can we just discard all of this sewer water in rivers directly? Or should we? Like, can we? The answer is yes, we can. And at many places it's being done also, which is not good. But should we discard them into the natural water directly?
No,
No, why?
because it could be toxic to the
It
particular
could be, it will be toxic. It will contaminate the water, because the river waters are also used for
drinking purposes.
drinking purposes. Correct. So we get fresh water from the rivers, and also that fresh water is used by humans to drink and to do all other activities, like to clean, like to use it for flushing, washing, etc., bathing, etc. And all of it goes into the sewers. Okay, and from the sewers, this sewage water should be properly treated before we throw it into the water, natural water body. So when I say treated, what kind of treatment are you expecting? What should I do? What should we do, people? What do we mean by treating sewage water?
We should make it less toxic.
Perfect. Because how can we make it less toxic? What is the idea? What is your idea of making it less toxic? Suppose I give this job to you. Jasola.
Decrease the toxic levels. Like
How? That's what I'm asking.
filtration, something like that.
What is the what are the toxics? What are the toxins that you are worried about?
Human excreta, harmful pathogens, bacteria, like that.
So how are you going to deal with it? Any idea? Of course, you'll put something into the water, right? That's is very obvious, correct?
So antibacterial and viral something medicines.
Antibacterial medicine. So you are going to, how much amount of sewage is produced by the cities, and to kill all the bacteria in it, how much antibiotic do you use? We don't have antibiotics to treat humans, and you want to put it in the sewage water to kill bacteria there. Sounds like a plan to me. What about you? Will you do that? And antibiotics are expensive. Would you like to use antibiotics which are expensive as comparatively to get rid of sewage? And even if you kill all the bacteria, there is still toxicity in that water. It's not such a difficult problem. You have to think. You don't have to do a science degree to understand. Okay, so what are you going to do? In that sewage, in that sewer's water, there is already many bacteria present, right? Many of these bacteria are pathogenic. Okay, and many other bacteria which may or may not be pathogenic but are heterotrophic, we will use the same bacteria and their ability to reduce the toxicity from the water, from that sewage water. Now, how are we going to do it? Write down. Let's write down its steps. Write down the first step. The sewage water, the sewage waste is treated in sewage treatment plants. In sewage treatment plants known as STPs, before releasing it into the natural water body. Okay. Now, this treatment is done by heterotrophic bacteria, which is already present in the sewage water. Okay. Now, this treatment have two stages. First stage is called the primary treatment, because apart from microbes, there may also be other bigger contaminations which we can just sieve out, right? Is the physical removal. You understand, everyone? So it's physical removal of large particles, like even small or large particles, like particles that can be sieved out. Okay, so it's a physical process. what we do in this process is we use filtration. So either things which are floating in water, you can filter it out using a sieve. So there could be like human hair. If this is present in the fecal matter, you can just sieve it out. Do you understand? Or sedimentation, things which sediment. So you just accumulate this sewage water and let it standing for some time. If it's standing, if it's not flowing, if it's just standing, so either things will float or things will sink down. So through both sedimentation and filtration, we can get rid of a large amount of particles. Is it clear?
Yes, sir.
Yeah. So from this we remove the floating waste, and then we can decide what to do with it. We can discard it, bury it in the soil, things like that. And with this, the sedimentation, all the solids that sediment, they make the primary sludge. The word primary sludge is used for everything that settles down. Okay. So this is what settles down. And the second thing that we get is the liquid. So that's called effluent. This is the liquid that, or also the supernatant, that does not settle down. Now this also contains a lot of microbes and pathogens, but at least we have, for the first round, we have figured out, we have removed floating waste through filtration. We have also removed the primary sludge and the supernatant. Now we have three things. This will get discarded. Sludge and supernatant is what we have to work with. Okay, now from this first treatment, you will take just the effluent, because finally we want to treat the water, right? You understand? So primary sludge also, whatever it contains, it's separated. So this effluent is taken out from primary settling tanks into secondary tanks. And here we begin the second phase, which is the secondary treatment. Now remember, the secondary treatment is happening for the effluent only, right? So it is also known as biological treatment. Is it clear, everyone?
Yes.
So it is happening for the just remember this thing, primary effluent only. Okay. Now first thing that we do with this primary effluent, like we did here, okay, is called aeration. Air is passed. Okay, so in large aeration tanks, there are aeration tanks where it is constantly agitated and air is pumped into it. Now why are we pumping air into primary effluent? Any idea? Constant agitation is done and air is pumped mechanically through pumps. You have seen, all of you have seen aquariums. So there's a pump at the bottom which keeps pumping air into the water, right? The bubbles keep coming out of the water. But there the water is standing still. We are not agitating it. So just imagine a rover, a blade at the bottom of the tank that is also constantly moving, mixing the water. And while you are mixing the water, agitating the water, which is the sewage water, you are also pumping air into it, just like you pumped it into the aquarium. So, but why it is pumped? What does air contains?
Oxygen.
Oxygen, right? So why are we worried about oxygen here? What are we doing with the oxygen? So aeration helps in what does it help in? Growth of useful aerobic bacteria. And what will it do? And will kill the harmful anaerobic bacteria. This is the first step. If you give oxygen to certain bacteria, they die. Okay. And these aerobic bacteria are known as flocks. The group, they form a layer. Okay. So flock is, you can write down, this has been asked in NEET. These are masses of aerobic bacteria with fungal filaments. So they look like a mesh which gets created over the water. Now, how are these flocks useful? Any idea? How are these flocks, these bacteria, useful? They are aerobic bacteria. Why are we increasing their number in the water, in the sewage water? Because sewage water, to begin with, had very less oxygen in it, right? So most of the bacteria were harmful, pathogenic, and anaerobic, disease-causing bacteria in the water. And this water's quality was very, very low because this water has very less dissolved oxygen. And there is something called BOD, biochemical oxygen demand. So if the BOD is high, the water is polluted. The more polluted the water is, the less oxygen it will have. Is it clear, everyone? Madiha, Memona?
Yes.
Yeah. So the sewage water, a fresh river water, the BOD will be very, very low. Okay, because it's fresh water, it has lots of oxygen dissolved in it. That's why fish and all sorts of life forms are living in it. But in a sewage water, only anaerobic and harmful bacteria live. So their BOD is very high. The demand for oxygen in that water is very high because there is no oxygen in that water. So these aerobic bacteria, what they do, they increase the oxygen level of water. And also, when they are growing, they consume major organic matter in the effluent. They need to eat something, right? So other organic matter they eat, and they increase the O2 level. Now due to increase O2 level, what happens? The BOD, which is biochemical oxygen demand, will decrease or increase? what will happen? For the effluent which is being treated by aeration, the BOD will increase or decrease?
Sir, can you repeat?
For the effluent, for the primary effluent which is being treated by aeration, the biochemical oxygen demand, the BOD, will increase or decrease?
It will decrease.
Why it will decrease? Madhya says it will increase. Why do you think it will decrease, Saifullah? Or Madhya, why do you think it will increase? See, these are the things you can either remember as sentences and add to your memory, or you can just understand the logic and realize, okay, next time whenever it is asked, I know. Samia says decrease. Samia, would you like to tell why? What is your rationale to think that it decreases?
Like you said that the more polluted the water is, the more the demand will be there. So if we provide oxygen, then the demand will decrease because it is getting oxygen already.
Exactly. When does the demand factor goes up? When there is less availability, correct? Right, Samia?
Yes, sir.
So to begin with, when the water was very polluted, that's what is the sewage water, it had almost no oxygen. No life could survive in that sewage water apart from pathogenic anaerobic bacteria. That's what we want to get rid of. So we started putting air in it mechanically. So that aerobic bacteria will now start rising up. They will start eating up the major organic debris and they will increase the O2 level in the water. It will increase the O2 level in the water. So when the O2 level, the oxygen level will increase, its biochemical demand will decrease. So BOD decreases in primary effluent. Now, if it is decreasing, can I say that this water is getting treated and is less polluted now? Yes? Yes or no? Can I say that a water with BOD which is less polluted, and the water with BOD which is more polluted? Okay. If I think you are not clear, so let's define this BOD. Write down. Okay, so me as clear, but write down what is BOD. Define BOD. A question, it can come as a 1 marker in your exam. Define biological oxygen demand. Write down. BOD refers to the amount of oxygen required by the bacteria, by the aerobic bacteria to oxidize all the organic matter present in it. All the organic matter present in it. And it is not just one type of bacteria, it's many type of bacteria. So basically a sewage water has lot of organic matter in it. So human excreta, nitrogenous wastes, cleaning, bathing, saliva, soap, all these things, waste from the skin, waste from the urine, fecal matter, from the kitchen, all of these have organic matter in it. Now, aerobic bacteria can degrade, destroy, metabolize all this organic matter and then convert it into simple chemicals which are non-toxic, like phosphorus, carbon, etc., etc. But to do this, these aerobic bacteria need oxygen, lots and lots of oxygen to oxidize it. Now, the demand for oxygen is to oxidize all these organic matter present in the water. So the more BOD means more oxygen is required to oxidize all the organic matter, which means there is lot of organic matter. Less BOD means less oxygen is required because there is less organic matter. So BOD is a measure for how much oxygen will be required by the microorganisms to make the water or the solvent free from organic matter and decompose all the organic matter. So it is a measure of organic matter present in the water. The greater the organic matter, the more polluted it is, and the greater is the demand for BOD. Okay, is it clear? Is it now clear?
Yes.
Yes. So this is step A, aeration. Now once we have done aeration, what we need to do? Aeration is done. The BOD in the sewage water has reduced a lot. Now what we want to do? Do we need this bacteria anymore? So we use these aerobic bacteria, the flocks, to just decrease the BOD. We give them oxygen by aeration and we allowed them to decompose every organic matter in the water. Now when all the organic matter is decomposed, do we need these bacteria? Can we throw the water in the rivers with all these bacteria? No, right? We need to get rid of this bacteria as well. So what we will do? Second is put. Now from here onward, we will call it the secondary effluent. Because secondary effluent have very low BOD. Okay. So we put this in settling tanks for days, for many days. Why are we putting this in settling tank? To let the flocks settle down. Okay. Is it clear? And this flocks which settle down is known as, so remember there was a primary sludge. This, what was it called? Here, sedimentation. This was the primary sludge which we removed. Now this, when this flock settle down, we call this as secondary sludge or activated sludge. They are like muddy substance, so that's why they are called sludge. Okay. Now, what is this activated sludge? Why do we call it activated? Any idea? Anyone? No idea? It's called activated sludge because the bacteria present in this sludge is capable of degrading organic matter in a given water sample, given that you supply oxygen in that water. So this activated sludge is very, very useful because some part of this activated sludge you can put back into the aeration tank to serve as inoculum. Just like to do curdling of milk, we put some curd in it, right? Containing the bacteria already, which we need in large numbers. So now instead of producing again and again the bacteria, we can just put this already activated sludge. So some part, write down, a minor part is pumped back into aeration tanks. Okay, to treat primary effluent. So this is done to keep this system working. But this activated sludge, what does it contains? Bacteria, lots and lots of aerobic bacteria, right? And
Yes,
fungi,
sir.
fungal growth. Now the major part, what can we do with this? Any idea? Instead of just throwing it, we can put it into large tanks called sludge digesters. Put into, now they are anaerobic sludge digesters. Now why are we doing this? We just wanted to treat the water, we did it. Why to worry about these sludge? Because where do we want to throw this? These are aerobic bacteria. Wherever we will throw this, they will start decomposing organic matter in that water or anywhere. So instead, they can be put in anaerobic sludge digesters. Here we use anaerobic bacteria. Okay, they kill and digest the aerobic bacteria. So we grew the aerobic bacteria in the first place to decompose all the organic waste, and because of which the anaerobic bacteria died. Now we are using more anaerobic bacteria to kill and digest aerobic bacteria. And remember, there were fungi as well, and fungi. Now what will happen because of this process? Because they are anaerobic, they cannot release oxygen. Now what will they release? During this digestion, what will they release? They will release methane, 3 gases. Majorly methane, which is CH4. They will release CO2, which is carbon dioxide. And they will release hydrogen sulfide, H2S. Majorly these gases are released, out of which this methane, can you tell me what is methane as a gas?
The biogas.
Yes, perfect. So methane has the potential to be used as a fuel. So now you understand why are we doing all this, killing the aerobic bacteria, putting it in anaerobic sludge digestion, using anaerobic bacteria to kill them and digest them to produce methane. Because this methane, so what happened to this part? So whatever comes from this secondary treatment plant, okay, before putting the activated sludge into the anaerobic sludge digester, the water that comes out, what we do it? So we can write down the effluent. So this secondary effluent, the effluent from secondary digesters or secondary treatment plant is released in the water body? Because it has no organic matter, it also has no aerobic bacterial load. Okay, so that water is released in the natural water body because it's very safe now. And what do we do with this gas, methane? So they form the biogas. So we got two benefits. One, we treated the water, made it non-toxic to be released in the water body again, and we also managed to produce biogas that can be used as a fuel source. Benefit or not? Is it clear, everyone? So sewage treatment is very important a part of this chapter. And what microbes help? What are these methanogens? What are these? So this bacteria that we talked about here in the secondary anaerobic sludge, this anaerobic bacteria, it contains methanogens. And what are methanogens? Methanobacterium is one methanogen which produce methane. Okay, in these biogas plants. Is it clear? Any doubt, people? Yes, no, something?
No, sir.
No. So please read this part from NCERT to get a more clear understanding. And if you have any doubts, ask me in the next class. In the next class, we will finish this chapter. The two parts which I am going to teach you is how to use microorganisms to make biocontrol agents, okay, against disease causing pests, especially in plants. Okay, and also the last part is using them as biofertilizers. That is small, but I will be talking mostly about BT cotton. So BT cotton is a genetically modified plant, and how is BT cotton produced? What is the benefit of BT cotton? That we will discuss in the next class. And we'll finish on this note and we'll start biotechnology, the application and principles of biotechnology in the next class. Is it clear?
Yes, sir.
Okay. So I'll see you all in the next class. If you have no doubts, I declare the class here. Take care.
Sir, can you scroll down?
Yes, sure, sir. I'm sorry. Let me share the screen again. Can you see the screen? I'll scroll down.
Yes,
Where
sir.
do you want me to stop?
Yes,
You want me
sir.
to zoom out a little?
No.
Is this fine?
Yes,
Can you
sir.
see it?
Yes.
Let me know when it's done, okay? Others can leave. Those who are noting and writing down can write it. Samia, do you understand something in today's class?
Yes, sir. It was pretty clear.
Okay. So this is not the best kind of chapters that we discuss in the class because it's still very factual. But next chapter, which is biotechnology, I'll be talking about the fundamentals and principles, which is very interesting a chapter. This is where engineering meets biology and bioengineers. So we are making technology, we are using technology for biological processes. Okay, so that will be a good chapter to begin with. Since you have joined a little late in the year, so what I would like you to do is please reach back to Learnivio, the number, and ask them for recorded video links of my previous classes. Okay, go through it, make notes,